Functional Traits and Ontology, part of the Systematics and Biodiversity course offered by Leiden University in 2020. I'm Jeremy Miller. In this lecture, we will explore the traits of species and the places they are found, and how these interact. We will consider how ontologies can be used to resolve ambiguity when discussing traits. We will see how the functional traits of the various species in a community contribute to ecosystem function, and how to assess ecosystem function using both on-the-ground methods and remote sensing. We all know by now that we are living in a changing world, and these changes in habitat and climate are impacting which species live where. Some species are expanding their ranges, sometimes taking advantage of human structures and practices to facilitate this. At a course level, we know quite a bit about the environmental conditions in different parts of the planet. The WorldClim database is publicly available historical data on global temperature and precipitation. It is based on a large network of weather stations collecting data. However, those stations are not evenly distributed around the world, and extrapolation for areas between stations in regions where the observation network is not dense or physical features are complex can be problematic. Satellite-based remote sensing platforms promise more reliable data because they are not dependent on the existing uneven network of physical observation stations, and they are more successful at dealing with physically complex features. Using knowledge of a species distribution and the environmental characteristics of those locations, we can develop predictive species distribution models. If we know where a species has been found based on specimen or observation records, and we know about the characteristics of those places, we can search for other locations that fit within the environmental envelope defined by those places. From this, we may be able to predict new locations where the species might be found. In some cases, there may be suitable habitats for a species far from their known range. This may suggest that if the species were somehow transported there, it would have a reasonable chance of establishing itself. In this era of climate change, it may be more relevant to apply climate change models, estimates of how temperature and precipitation might change across the surface of the Earth in the future. The shape of that environmental envelope, based on current species records, might be predicted to change. It might expand or get smaller. It might shift to the north or south, or be displaced to areas of higher elevation. The niche that a particular species occupies is defined by a number of factors. Climate is an important one. Different species have different competitive strengths, at least at some times and places. But competition, interactions with predators, parasites, diseases, and conspecifics, and dependencies on other species, such as is the case in pollination systems or symbiotic relationships, or on physical features like substrate, are also important for characterizing a niche. So a species may not be actually present in the entire area in which the climate is suitable, due to missing dependencies or competitive exclusion, or simply lack of opportunity because propagules have not dispersed there yet. Phenotype is the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. Observable characteristics may include the organism's morphology, its physical form, its development, behavior, biochemical and physiological properties. Variation in phenotype is the raw material that drives evolution and is also the basis of taxonomy. Taxonomy is about breaking down elements of phenotype into comparable units, describing elements of the phenotype of each species and contrasting that with other related species. Similarly, phylogenetic inference typically depends on building a matrix of taxa by traits. It is important that traits are putatively homologous, that is, characters with shared ancestry. One example of this would be the forelimbs of tetrapods, which take many forms and functions: walking legs, fins, wings, and the multi-purpose utilities of bipedal hominids that are our arms. Traits on trees can form the basis for testing hypotheses about how traits interact with each other and can reveal trends over space and time. An ontology is a system of controlled vocabulary terms and definitions. Language is often ambiguous. Words can have more than one meaning. When talking about species traits, we want to be unambiguous about what we are describing. So let's take a non-biological example, the word mercury. Mercury can refer to the planet in our solar system that is closest to the sun. It can also refer to a chemical element. The periodic table element code for mercury is Hg, so this is a synonym of the chemical element mercury. Mercury is also the name of a figure from Roman mythology and the name of a popular model of automobile made by the Ford Motor Company from 1938 to 2011. If we want to describe mercury or compare mercury to other similar things, we have to know that our use of mercury has the appropriate relationship to other terminology. We might want to compare mercury to other planets, but it would be nonsensical to compare planets to a chemical element. So let's now think of ontology in the context of species traits, taking as our example the Hymenoptera anatomy ontology. Hymenoptera are the sawflies, ants, bees, and wasps. They are a megadiverse taxon with more than 115,000 species described. The Hymenoptera anatomy ontology extracted terms from taxonomic treatments, phylogenetic studies, and accounts of functional morphology. Problems of anatomical terminology are similar to problems with taxonomic names. A single term may conflate several non-homologous traits, or there may be multiple synonymous terms for a single trait. The resulting ambiguity leads to miscommunication and wasted effort. The solution is a system of controlled vocabulary. For example, let's take this structure, the paired claws at the tip of the leg. A survey of literature on Hymenoptera found more than 1,500 synonymous terms for this structure. But the ontology maps relationships among terms, and it knows that all these terms point to the same homologous structure. So we can run a query for any one of these terms and return results that contain any of the synonymous terms. But synonyms are a relatively easy problem to deal with. The more difficult problem is homonymy. Here is an example based on the term paramere, which has been used in the literature to refer to at least 5 different and incompatible sets of structures. Because of this, there is no straightforward way to query for just one of these definitions of the term paramere. But the Hymenoptera anatomy ontology has designated a unique identifier for each of these concepts, and this does give us a basis for forming queries. Once we have a broad sample of characters scored across Hymenoptera, we query the database to find taxa with particular combinations of traits. This can be useful for identification or to study the evolution of particular traits. Literature on biodiversity is a rich source of trait data. This generally comes in 2 forms, either as structured tables or unstructured natural language text. Structured tables are typically found in phylogenetic studies. The cells where any taxon and character intercept is where the particular character state is represented, which is described with text and may also be documented with images. Structured tables are relatively straightforward to use as trait data because ontology and phylogenetic character matrices are both concerned with homologous characters. This schematic outlines the key elements involved. We begin with specimens. Taxonomy tells us which specimens belong to each taxon and how that taxon fits within the scope of our study. Comparison of the various homologous anatomical structures reveals variation that can be scored as contrasting character states. Ontology helps to structure the matrix based on homologous traits and unambiguous terminology. Images taken from standard views to facilitate comparisons document these traits. Structured trait data can also be extracted from unstructured or semi-structured text. Taxonomic descriptions are often written in a kind of parallel structured language. They cover characteristics in a particular order and use the same language in descriptions of different species when the trait is the same, and different language when it contrasts. Here are 3 brief excerpts from a taxonomic publication on a group of spiders. The description specifies the relevant part of the anatomy, followed by a description of some aspect of it. In this case, the coloration of the carapace is described. Next, the texture and physical features of the carapace is described. This continues for various other structures, the sternum, the legs, and the abdomen. There are efforts to build natural language processing algorithms to convert semi-structured text, such as you would find in a taxonomic description, into structured trait data based on ontology. Now we are going to focus on a particular class of traits called functional traits. These are characteristics of an organism that influence performance of fitness and may be morphological, chemical, behavioral, etc. The big idea is that an ecosystem function is the product of the functional traits of the various members of its biotic community, scaled by their abundance. Conceptually, this idea has been around for a long time, but we have only recently begun to see rapid development and practical applications of this idea. Studies of functional traits can characterize community responses to environmental change, or quantify the influence of community shifts, such as those predicted under climate change models, and thereby predict shifts in ecosystem function. Nevertheless, what exactly constitutes a functional trait is not straightforward in practice. We are looking for traits that are consequential for ecosystem function, but which traits are these exactly? How can such traits be measured or inferred? Are the traits being considered independent of each other, or are some positively or negatively correlated with each other in a way that might distort your interpretation of the data? How many traits are needed, and how should they be selected? What is the most appropriate framework or statistical approach for testing the data? Functional diversity is the value and range of those species and their traits that influence ecosystem function. Think of it as functional traits across all the various species in a community, scaled by their contribution to the overall community. Altogether, this adds up to ecosystem function. So we can look at the various species in a community and their contributions to ecosystem function today, or we can make predictions on how ecosystem function might change as the organisms making up that community change in both kind and abundance. Functional trait studies have become particularly important in the world of botany. Plants vary in all kinds of comparable ways, from the way their leaves function to deal with sunlight and water, their relationship with various nutrients, and their persistence in the environment, from short-lived herbs to long-lived woody trees. All of these factors can have consequences for things like erosion, nutrient cycling, and temperature. Traits can be measured empirically or extracted from trait databases. One example is the TRI database, a global database of plant traits focusing on growth, reproduction, dispersal, establishment, and persistence, aggregated from various smaller plant trait databases. Let's take one example of a study linking plant functional traits to ecological processes, investigating the functional consequences of biodiversity loss. This study by Gross et al. 2017 found that the abundance distribution of two functional traits, specific leaf area and maximum plant height, had a stronger impact on multifunctionality of ecosystem function than other factors, including proxies for nutrient cycling, biological productivity, soil fertility, and species richness. This suggests that existing levels of ecosystem function can be maintained even in the face of species lost, at least for the domain investigated here. The approach I've highlighted so far involves measuring or otherwise assessing the traits of organisms, then extrapolating upwards to obtain a value for the community as a whole. An alternative approach is exemplified by remote sensing. This might be a satellite network, but can also involve other kinds of sensors. Remote sensing offers various trade-offs with the ground-based approach. It does not require taxonomic assessment, which makes it equally applicable everywhere, regardless of the state of taxonomic knowledge. It can accommodate interactions among species that might not be apparent from the ground-based approach. It can deal with rare as well as common species. It is, however, limited to what can be observed remotely. In one important validation study, a series of functional traits were measured both remotely and on the ground, and produced similar values. This suggests that we can use these two alternative approaches as best suits the particulars of our study question, yet have confidence that both approaches are converging on the same signal. So we'll end this presentation with this schematic slide that you have seen previously. Keep in mind that knowing which species live in a place is just one aspect of biodiversity knowledge. It is the functional traits of the various species, scaled by the magnitude of their presence within the community, that produces ecosystem function. Arguably, it is the risk to the loss of ecosystem function that holds the most risk for our own species as the biodiversity crisis continues to unfold.